Okay, so now we're going to discuss phagocytic cells in a little bit more detail. So keep in mind that we have two truly phagocytic cells. We have our neutrophils and we have our macrophages. And to be a truly phagocytic cell, you have to want to actively eat something and you have to want to actively try to break down something and neutralize it. And so that's what neutrophils and macrophages do. And so the phagocytosis, or is a process of internalization of matter outside of the cell into an endocytic compartment on purpose. So it is active eating. And so pathogens can be a trigger, dying cells can be a trigger. Opsonins are going to help. Okay, so we have our C-reactive protein, we have our complement C3b, and we have our IgG are going to be our opsonins. And then, of course, your phagocytosis and your phagocytic cells are going to be our alarm system. Especially our macrophage. And this is going to release cytokines to alert and chemokines to direct. Now, in the process of trying to kill the pathogen, there are several ways these phagocytic cells do this. They can use their lysosomes, and they have digestive enzymes in those lysosomes. They can use an oxidative burst. And newts have nets, which we'll talk about. And so these phagocytic cells, of course, are important in extracellular bacteria. And they're also important in our defenses against intercellular bacteria, such as Mycobacterium tuberculosis, for example. And so let's discuss them a little bit more. So I'm going to talk first about neutrophils. So neutrophils are known as a granulocyte. They do have this multi-lobe nuclei. And so they're similar to our eosinophils or basophils in that respect. Their cytoplasm is clear. They don't have the granules that eosinophils and basophils have. At least they don't stain. They do have granules, but they're neutral granules, whereas basophils are going to stain purple because they have acidic granules, and eosinophils are going to stain blue because they're going to stain red because they have basic granules. Now, a neutrophil is, again, the most abundant white blood cell in the actual body. You don't find them circulating that much. You will find some, but not very many. They're going to be stored in the bone marrow, and they're stored in large numbers. They're stored there as mature neutrophils waiting for a signal to be released into the blood, and they get released in mass. They go out there and they try to find where they need to go and destroy whatever is causing problems in the body. So what triggers their release is a series of cytokines. We have IL-17. We also have IL-1 via IL-17. What that means is IL-1 is an acute phase response, but IL-1 itself cannot release neutrophils, but it can activate IL-17, which then will cause the release of neutrophils. Now, the neutrophils are going to traffic to the site of infection. And so there are a few proteins we can introduce right now. We have glycoproteins on the surface of the neutrophils that are going to bind to E and P selectin on an inflamed endothelial cell. So this endothelial cell has upregulating sticky fingers, the EMP selection. The neutrophil comes rolling around and has a glycoprotein that responds to that, so it kind of tethers itself to that very loosely. You then get a more rigorous interaction through ICAM1 on the neutrophil and ICAM2 on the endothelial cell that's inflamed. And this stabilizes the reaction. Remember, ICAM is intracellular adhesion molecule, and it's important for stabilizing cell-to-cell interactions. We saw ICAMs when we were talking about our antigen-presenting cell and our T-cell interacting, so it's the same molecules. But in this case, we're on the endothelial cell and the neutrophil. And the endothelial cell is really trying to tell the neutrophil, this is where you need to stop, and this is where you need to enter. And so the neutrophil will enter, and it'll do this through that process we've talked about already called diapedesis. It squeezes through your endothelial cells, and then it will be recruited to the site of the bacteria through some gradient. And here we have complement C5a, which is an anaphytoxin that is causing a gradient that the neutrophil will use in order to find the bacteria. Your neutrophil will kill the bacteria, and your neutrophil will always die and generate pus. Regardless of what tissue you're in, you will always have pus formed. Now, the neutrophils are critical for defense, and a patient that is neutropenic are at risk for bacteria infections and sepsis. Now, neutropenic means that you have immature neutrophils that are in the blood and less of them. Now, again, neutrophils are very important for extracellular bacteria and fungi. And if you are neutropenic, you're not able to control those infections. They can get out of control very quickly and lead to sepsis and shock and death. It's very hard to treat shock, as we're going to find. And so one way to avoid that is to try to keep people not neutropenic. And so that is one to monitor people who have a loss of neutrophils for whatever reason it might be. Okay, so now let's talk a little bit about our monocytes and macrophages. So one thing to keep in mind, a monocyte is in our blood, and a macrophage is in our tissue. So when a monocyte moves from the blood to a tissue, it becomes a macrophage. They're kind of the same cell. So a macrophage is our tissue resident, and our macrophage is long-lived. They help to maintain homeostasis. They are the alarm system. They protect against non-virulent microbes. If a macrophage can eliminate the microbe on its own, we don't need to call in our T cells, B cells, etc. Macrophages are really good at eating things and getting rid of them unless the pathogen or the microbe has developed a way to get around the macrophage's defenses. It is, of course, an antigen-presenting cell. It, of course, expresses, it has MHC1, MHC2. It has a co-stimulating CD80, 86, also referred to as beta 7. Macrophages also have CD40, so they are going to require help, so they can get help from your CD4 T cells. Monocytes circulate at a steady level and then enter tissues. And in the tissue, they become the macrophage. Okay, so just a comparison of phagocytic cells. So here I have a list, it's in your book, but some things to point out is neutrophils are going to be continuously produced, and it increases an infection. And that's different than your macrophage, because your macrophage is just produced at a steady state. There's no increase due to infections. Neutrophils are always going to be stored in the bone marrow. Your macrophages and monocytes are going to be in the blood and tissues as resident cells all the time. Neutrophils have to migrate to inflamed tissues, whereas your macrophage is going to be a resident in tissues. Neutrophils have only one mature form. Macrophages have various. You can have an M1 macrophage, M2 macrophage. M1 tend to be more pro-inflammatory, and M2 tend to be those found more in areas where you don't want so much information, such as your gut, for example. They both are going to do phagocytosis, so there's nothing different there. They both have phagocytosis. They are both going to kill via toxic molecules, granules, lysosomes, oxidative burst. Your neutrophils have nets. Your macrophages have no nets. They're both going to be able to recruit more neutrophils. Macrophages don't really get recruited. They have differences in how they respond to T helper cells. So your neutrophils will respond to IL-17 from a Th17 helper cell, and your macrophages are going to be more pro-inflammatory, so they're going to be more involved with Th1 cells. Your neutrophil doesn't really interact with the adaptive immune response, whereas your macrophage is an antigen-presenting cell, so it interacts a lot with your adaptive response. So now just a little bit on the types of macrophages. Remember, there's only one type of neutrophil. There's many macrophages. Tissue macrophages are found in many tissues, and these are your resident macrophages. They're going to be large cells with granules. That is a big one. They are the typical macrophages. If they're in the bone marrow and lymph node, they're called histocytes. Histiocytes. Okay. We also have giant or epithelioid cells, and this occurs during chronic inflammation. And the macrophages are going to form multinucleated giant cells. They're also called epithelioid cells. And basically, the macrophages fuse with one another, so they become giant. This is going to be driven by your T helper cells. Mostly a Th1 environment, and they're going to form granulomas during prolonged responses, such as during tuberculosis infection. And we'll talk about what a granuloma is. Granuloma is basically a group of macrophages that fuse into giant cells, and their center is so oxygen deprived they actually have an area of necrosis. And it's in TB, it's actually that area of necrosis that people cough up and they can spread the bacterium. So that's active TB. We have our fixed macrophages, and these are going to line the sinusoids of the spleen and liver. You have various types of macrophages in the spleen. You have your marginal zone macrophages, red pulp macrophages, all kinds of macrophages. And in the liver, of course, they're called Kupffer cells. And they're going to phagocytize circulating matter and dying cells, so they are a filter. Your red blood cells are usually eliminated in the spleen, but your Kupffer cells can also pick up dying red blood cells and digest them as well. We have our alveolar macrophages, which are going to be an innate defense system of the lungs. And these are going to participate in chronic obstructive pulmonary disease, for example. Okay, and then we also have purple glial cells. Glial cells are long-lived nervous system macrophages. And remember, our nervous system, such as our central nervous system, is going to be a privileged site. So their main role is homeostasis. They're going to clear cellular, neuronal, dead cellular debris. Now, of course, if this brain or the central nervous system does become infected, they will participate in that as well. But on a day-to-day basis, they are there to maintain homeostasis in the brain. And then we also have our osteoclasts. Let's do this one. Okay, our osteoclasts are the most specialized. And they're going to regulate calcium metabolism. This is going to be done by reabsorbing. Remember, they're macrophage, they eat. Of bone, and then they release the calcium into the blood. So that's an easy way to remember what they do. They are macrophages, so they actively eat. So osteoclasts are macrophages, so they're going to eat. And so they eat the bone, reabsorb it, and then they're going to release the calcium. Okay, so recruitment of neutrophils. So again, here is just another drawing that I did about the whole process of diabetes. So we have, again, our neutrophil that is going to be recruited via IL-17, for example. So we can say we have somewhere in the bone marrow. Here's draw bone. Here's our bone. And then we have our marrow. Oh, actually, okay. Let me do that red. So we'll have our bone marrow. Okay, and then we'll have our IL-17. So we have a Th17 cell in the gut, makes IL-17, and then you release your neutrophils. Okay, so your Th17 will release IL-17, or your macrophages can release IL-8. So that's another thing that can come. And so your neutrophil will be released from the bone marrow. It's going to go and interact with your actually I should put it over here, huh? Because this is our, sorry about that. This is our inflamed side. Okay, so now we have our newts. So normal tissue we're showing here, we have our macrophage. And our neutrophil passes it by because there's nothing telling the neutrophil to stop. Macrophages have different ways of getting into tissues. Our neutrophil finds an inflamed endothelial area, and this is where you're going to have the E and P selectin on your endothelial cells, and they're going to bind to glycoprotein ligands on the neutrophils. And then you're also going to get more adherence through ICAM2 on the endothelial cells, interacting with your ICAM1 on your neutrophil. And your neutrophil will be tethered more tightly at this point, and then the neutrophil will enter the tissue. So again, the inflamed tissue is going to express E-selectin and P-selectin, those sticky fingers, as well as the ICAM. And then your I guess I should go through this. So your macrophage ate your bacteria, released all of the acute phase response cytokines, and then this activates the endothelial cells. It express E-selectin, P-selectin. You also have complement is getting activated. C5a, for example, is causing a gradient. You have your neutrophil that's going to bind diapedese, and then it's going to find its way via the C5a gradient to the site of infection, and then your neutrophil is going to help to eliminate the actual pathogen. Okay, so phagocytic cells and their receptors. So there's going to be various receptors. We have receptors that are important for migration. And these are going to be the integrins. And you're going to have chemokine receptors. Those are G protein receptors, similar to epinephrine. We also have the need for migration is going to be during infection. Macrophages can enter non-disease tissue. So let's see, migration, we have our newts, infection, oops. So you have your E and P selectin, and then you have your ICAM 1 and 2 interactions. And for migration for our macrophages, remember macrophages enter tissue just normally. Macrophages use Y6C and LFA1. And then we also have our pattern recognition receptors. Those are PRRs. And so this is going to be our toollike receptor. We have our C-lectin receptor. And this will bind to sugar. And it can bind to either a pathogen or a dying cell. And your phagocytic cell have receptors that bind to C-lectin and ingest whatever the C-lectin is attached to. We also have complement receptors. So we have our C3b opsinin. We have our immune complexes. And we have dead cells. We also have our immunoglobulin, FC region. So we have IgG, for example, FC gamma R1 will bind to IgG. Now, I'll talk a little bit about toll-like receptors because there's so many of them. We have different toll-like receptors. You can see that we have toll-like receptor 2, 3, 4, 5, 7, and 9 listed here. One thing to keep in mind is that our toll-like receptors that are going to be binding to nucleic acid, so we have double-stranded RNA, single-stranded RNA, as well as cytosine guanine sequences, CpG. And so all of these are going to be on endosomes. So we have our TLR3, 7, and 9. These are going to be in the endosome compartment. Toll-like receptors are always membrane bound. And then we have our other toll-like receptors. We have toll-like receptors that recognize sugar and lipoproteins on the surface. Oh, that's not really too different. LPS, which is on the surface, flagella, which is on the surface. And so toll-like receptor 2, 4, 5 are going to be on the cell surface. And that's because your macrophage will see a flagella on an extracellular bacteria, but it won't see the RNA and DNA of a virus unless the virus is internalized. So that's why those receptors are in endosomal compartments. So there are a wide variety of cells that express these. Macrophages, you can see, are going to be important in many of these, as well as dendritic cells. The pathogens, again, the ones that are associated with our extracellular are going to be bacteria primarily, bacteria, and then the ones that are associated with our nucleic acid are going to be our intracellular bacteria and viruses. And so just make sure that you know these. I'm not so interested in what it's expressed on, but definitely the ligand, the pathogens, and the actual toll-like receptors. Now, drugs are being formed to bind to the toll-like receptors. And this is to induce inflammation. And so this is used to boost a vaccine. This is an adjuvant. So we can use CpG motifs. Binds to TLR9. And that's going to promote Th1. And so that would be important for vaccines and cancers. You can have equim mode. Let me say that wrong. It's another drug. And this mimics single-stranded RNA. And so this is TLR7. And this is used to treat warts. And this is, again, TLR7 is going to stimulate danger type of signal. It's going to recruit inflammatory responses, etc. Okay, so let's talk about functions of phagocytic cell. There are a lot of functions, and they are very complicated functions, not so easy. This is just one slide, but I would encourage all of you to go back to your books and look at all these. So phagocytosis, when you think of a phagocytic cell, you always think of phagocytosis. And remember, this is active eating. And it's going to be most effective if an opsonin is used. And typically the opsonins are going to be C3b or an immunoglobulin IgG. The phagosome is going to do exactly what it does when an endosome is taken up. It's going to fuse with a lysosome, and then you're going to kill the pathogen. Now, one way that you can kill pathogens is through something called an oxidative burst. So after phagocytosis and things like T cell help, if you're a macrophage, it's going to act. Your oxidative birth pathways are going to be activated, and the macrophage is going to produce toxic molecules to kill the pathogen. So we're going to have O2 minus hydrogen peroxide or myeloperoxidase, which will then become hypochlorous acid. You also can get inos, nitric oxide. And when too much is produced, you can decrease vascular tone and decrease cardiac output and decrease blood pressure, which is associated with septic shock. They also have many proteolytic enzymes. So again, you have the lysosomes within the endosomal compartment, which the phagosome is part of. And so you're going to fuse the phagosome with the lysosomes, which has those nice digestive enzymes. In the neutrophil, you're going to have granules that are going to be released. So that's a little bit different. So we have our macrophage, which you have phagosome to lysosome. And we have our neuts, which you have granules. Let me just put degranulation. So whereas the macrophage kind of wants to stay alive, the macrophage wants to eat things and digest things, the neutrophil is like, it just wants to explode and kill everything around it, whether it's going to kill bacteria or its own cells. It just releases all these toxic granules. So inside of these granules, you're going to have proteolytic enzymes that are going to digest the bacteria. You're going to also have defensins, which are peptides. So you can have, within the lysosomes, you can have defensins, which are peptides. You're going to have proteolytic enzymes in both of them to digest. You're going to have serpins. And serpins are going to protect the body from the enzymes that are leaked out of the lysosomes, such as alpha-1 antitrypsin. Now, for a neutrophil, there's also the ability to form nets. So this is a neutrophil only. And so basically what happens when the neutrophil is overwhelmed, the neutrophil will basically break down its cell membrane and release its chromosomes. Now the chromosomes are made up of DNA and histones. It's negatively charged, and so it gets very sticky, and it's going to sort of throw a net over the bacteria in the vicinity and trap the bacteria. The bacteria are not going to be able to divide. They're not going to be able to get nutrients. And in addition to trapping the bacteria, these nets also accumulate all the toxic things the neutrophil released. And so it's going to start to digest the bacteria as well. And so the nets are sort of a last resort for the neutrophil. And the neutrophil obviously is going to die at this point. So one side note is that the DNA can be an antigen. And may be a source of antigen in systemic lupus erythematosus. Which is SLE. You also have signaling. So we have prostaglandin and leukotrienes. This is our LOX. And so they have the ability to make prostaglandins very pro-inflammatory. And again, those are going to be important in promoting more inflammation. You also, with respect to these, you have your neutrophils, which are pyogenic, pus-forming. And then you also have macrophages that can further cause inflammatory responses to develop into granulomas. Granulomas are chronic. And we've already talked about the acute phase response, so just keep in mind acute phase response is our macrophage, and it's going to make the IL-1, IL-6, and TNF-alpha. It's going to trigger the fever response in your hypothalamus through your prostaglandins. It's going to increase metabolic rate, anorexia, weight loss. It's also going to recruit newts via IL-8. Okay. All right, so we just have a couple more here. We have to talk a little bit about defects. So we have chronic granulomatous disease, which is caused by NADPH oxidase systems being defective. So this is a primary immunodeficiency disease. And why we call it a primary is because this is at the level of the genes. A secondary immunodeficiency disease, which we'll talk about later, is something that's caused by an infection such as HIV. But if it's a primary immunodeficiency, it means there's a genetic defect. In the case of chronic granulomatous disease, it's a defect in NADPH oxidase. So a person with your NADPH oxidase deficiency cannot produce superoxide radicals or kill pathogens. And so you have recurrent bacteria plus fungal infections. There is also neutropenia, which is a decrease of neutrophils, and is usually caused by drug treatments. And it can lead to an increase of bacteria plus fungal and sepsis, because you really need your newts to get those guys under control, or else they overwhelm your system and you lead to sepsis. Now let's just go over a very quick overview of the recognition of pathogens. In our adaptive response, we are going to be using our T cell receptor associated with MHC. We're going to be using our B cell receptor to identify pathogens directly. So we can detect millions or many more millions, 10 to the 18th. It also sees self versus non-self. Not foolproof though. And so you can get autoimmunity. Innate, it recognizes danger. And it's going to be through those pattern recognition receptors that we've talked about, toll-like receptors, complement receptors, etc. So those would be danger signals. It releases cytokines. It's going to release co-stimulatory molecules and danger signals. And it's the alarm system. Okay, and so that's the end of this lecture.